In this screencast, we're going to look at comparing the processes of mitosis and meiosis, looking at some similarities and some differences. So the main difference in comparing mitosis and meiosis is that mitosis conserves the number of chromosome sets, and it produces cells that are genetically identical to the parent cell and to each other. Okay? Mitosis reduces the number of chromosome sets from 2 sets, which is a diploid cell, to 1 set, which is a haploid cell. Okay? The cells produced from a meiotic division are different genetically from each other and from the parent cell. So here is a comparison of chromosome movement during mitosis and meiosis. If we start both processes with a parent cell that is diploid, if we look in mitosis, okay, they all undergo replication of DNA during S phase and then prepare to enter mitosis. Mitosis goes through 1 division: prophase, metaphase, anaphase, and telophase. Okay, and results in 2 daughter cells that are both still diploid and genetically identical to each other and to the parent cell. In meiosis, we take our parent cell, go through S phase where we replicate the DNA, and then we have 2 divisions. In meiosis 1, prophase, metaphase, anaphase, telophase 1. First, in prophase 1, we have the homologous chromosomes pair up in synapsis. Then they recombine and exchange genetic material. The points of recombination are called chiasma. And then the homologous pairs line up during metaphase. And the homologous pairs separate during anaphase 1 and telophase 1. Okay? The results at the end of meiosis 1 are 2 haploid cells. Okay? And so they're already genetically different from each other. Then, over the course of meiosis 2, the sister chromatids separate, and the result is 4 haploid cells that are genetically different from each other and from the parent cell. Okay. If we look at a couple of key events, like DNA replication, in mitosis, this occurs during interphase, during S phase, before mitosis begins. And in meiosis, it occurs during interphase, again, S phase, before meiosis 1 begins. Number of divisions, there's 1 cell division in mitosis and 2 divisions in meiosis, and each include the 4 main phases. Okay, synapsis of homologous chromosomes does not occur during mitosis, and in meiosis it occurs during prophase 1 and is accompanied with crossing over between non-sister chromatids, resulting in chiasmata that hold the homologs together. Number of daughter cells. From mitosis, you have 2 daughter cells, and they are genetically identical to the parent cell and to each other. In meiosis, there are 4 daughter cells. They are haploid and are genetically different from the parent cell and from each other. Roles of mitosis in the animal. Multicellular adult to rise from a zygote, cells for growth, repair, and in some species, asexual reproduction. For meiosis, the role of meiosis is to produce the gametes. Okay, meiosis is a reduction division. It reduces the number of chromosomes and introduces genetic variability via crossing over and recombination, among other things. Okay, 3 events that are unique to meiosis only, and they all occur in meiosis 1. Okay, the first one is synapsis of homologous chromosomes and crossing over during prophase 1. Okay, homologous chromosomes physically connect and exchange genetic information. Second event, so that's one event that's unique to meiosis. Second event, at the metaphase plate in metaphase 1, there are the homologous pairs line up in their tetrads instead of individual replicated chromosomes. And then the third event is at anaphase 1, the homologous chromosomes separate. So sister chromatids do not separate, homologous chromosomes separate. And this is where we reduce, reduce the number of sets of chromosomes. Okay. And then just a few last endpoints. The sister chromatid cohesion that keeps sister chromatids together. Okay, that stays connected throughout meiosis 1 and holds those sister chromatids together. These protein complexes called cohesions are responsible for this, cohesins. In mitosis, the cohesins are cleaved at the end of metaphase. And in meiosis, cohesins are cleaved along the chromosome arms in anaphase 1, and this allows for the homologs to separate. And then in the centromeres in anaphase 2, and this allows the sister chromatids to separate. Okay, and that's it for our comparison of mitosis and meiosis.